Some of the elements of this lecture will be review, kind of reinforcing important concepts from previous lectures. Some of it will be a little bit new. We're going to be fairly general for this lecture. In the lecture for the next part of this unit, 2.2.2, I think it is, I will go into specifics on insects and talk about what's known generally about the origin of the insect body plan. Okay, so reminder, these toolkit genes, which remember are the morphogens, so usually mRNAs, but sometimes the protein products of those mRNAs are being created in a small group of cells called organizers, and then they are diffusing out across gradients to have an impact on target cells close or far away, depending on those threshold levels of activations. And they do that in one of two ways. Either they themselves are transcription factors. So a transcription factor is a protein that binds to the genome. It binds to DNA. And there are a couple of different motifs, portions of the protein, that bind to DNA and allow it to do its job. These are 4 different types of DNA binding motifs that we find in proteins. All of them are used in different toolkit genes. In animals, the most common one is this helix-turn-helix formation. There are actually 3 helices and 2 turns, and this fits into the minor groove of DNA at a specific sequence of DNA. So depending on the amino acids that are present in these helices, it's going to bind to DNA. And then other parts of the protein interact with cofactors and other things to then turn on and activate the genes where they are binding. So a transcription factor, if it's in an mRNA form, needs to get into its target cell, be translated into the protein, and that protein needs to get into the nucleus and bind to the DNA to have its effect. If it's already present in a protein form, then it just has to get into the nucleus to actually work and bind to the DNA and then activate genes and kick those genes down a different developmental pathway. Now, the second option, this is a diagram of a dimer. It's actually two of these helix-turn-helix that are bound together, and each one binds at a different place, giving it a little bit more specificity. Okay, but again, they're going to interact with very specific sequences of DNA, and so they will bind to some and not others. So the second option, instead of being a transcription factor themselves, these toolkit genes can be signaling molecules or part of a signaling pathway. So they can be the ligand. This protein can bind to a cell surface receptor and then catalyze a reaction. Typically, this is through phosphorylation or some other series of chemical reactions. Often there's a cascade and there can be multiple downstream targets. But even these, eventually, we have activation of a different transcription factor. There needs to be a transcription factor involved somewhere. So either the genes are transcription factors themselves, or they are parts of a signaling pathway that eventually will reach a transcription factor and bind to target DNA and turn on, or sometimes turn off, but regulate the expression of these target genes. Okay, now, originally, the Hox genes, these really important control genes for anterior to posterior patterning, were discovered in the Drosophila. But very rapidly, as researchers began to explore other organisms, they began to be found in other metazoans, including vertebrates. All chordates have them. Okay, so there are orthologs of, remember that term ortholog means if we traced back the ancestry of A1 in a vertebrate, labial in a Drosophila, we trace them back to their common ancestor, there would be a single gene in that ancestor that gave rise to those 2 orthologs. And remember, all these genes are results of gene duplications. They have enough sequence similarity that we can identify them as all part of the same gene family. So they are all paralogs of one another in Drosophila. And so there are multiple orthologs and paralogs. We're not going to do it in detail, but in a later lecture, we'll look at the more complex Hox clusters. There are actually 4 of them that are present in vertebrates. And so we can, there are only 3 species here, but now we have many, many more species of animals. We can infer what the ancestor of, for instance, all Bilateria, what did its Hox cluster look like? And even though it's long extinct, and even if we found fossils of it, there's no way we'd be able to look at its genes or analyze its Hox cluster. But we can do this through phylogenetics and mapping the characteristics that are present in modern organisms onto a phylogeny and figuring out what the ancestral characteristics were. And so as we've done this, we've found that Hox genes are present in all animals. And we're actually going to identify some key innovations in the Hox genes at a later date. And we'll map them. There are three of them that you need to know. We'll map them and talk about them when they occurred. But for now, just know that we find Hox genes in all animals, sometimes many, many dozens, at least 3 or 4 dozen, and sometimes just a handful. And in some of the simpler ones, so here we've got only the bilaterians, which have fairly complex Hox genes. As we move down into other animals, so if we look at the cnidarians, right, the jellyfish and their relatives, if we look at the sponges, we will find Hox genes, but they're much simpler. So an early simple group of Hox genes, if you want to call almost certainly a Hox cluster, was found in the ancestor of all animals. And then in the bilaterians, it begins to become more complex and more important in establishing this basic foundational pattern for the bilaterian animals. Okay, but Hox genes are present in all of the metazoa. And of course, if we mutate these Hox genes like we did in the fruit fly, if we mutate them in other organisms, we get a similar type of mutation. Now, one of the interesting things, this is one of the ramifications of having multiple Hox clusters in vertebrates, is that often there is redundancy. So there's an overlap in function of some of the duplicated versions of Hox genes. So this creates an interesting range of phenotypes that are possible for Hox gene mutations in the vertebrates. So here's a wild-type mouse embryo with 6 of these sacral vertebrae. Those are the vertebrae in the lower back. And that's very standard. Mammals all have a fairly standard pattern. There's various levels in the thoracic vertebra. But within a species, it's always the same. And if it's not, it's usually a fairly severe negative mutation. So here we have a mutant version that is missing 3 of the 4 copies of the Hox gene that's responsible for patterning these sacral vertebrae. And we end up with a duplicated sorry, I just had a little mental fade out there. A duplicated module, right? These modules of these repeated bones and associated structures that make up the vertebral column. So we ended up with a duplicated one. Now, if we have a quadruple knockout, so notice that even though we only have 1 functional copy of this Hox gene of the 4 that are possible, we still get only a minor phenotype. Still probably pretty major for the organism. There'd probably be some moderate to severe phenotypic impact for this. But if we knock out all of them, then we get even more defects and we get even more additional repeated segments. So this is kind of cool because vertebrates, by duplicating their Hox gene cluster, have a little bit more resiliency to mutations in them. If we have one that mutates, then the other 3 may be able to compensate. And it's not until we get 3 mutations that we start to see a phenotype. In this example, it could be more or less, but there's a little more flexibility, more range in vertebrates because of the redundancy that's built in via their Hox gene cluster duplication. Now, this presence of these basic patterning genes across all metazoa came as a big surprise. Again, our hypothesis going into the study of the genetic basis for animal form and animal evolution was that different genes in different organisms will have arisen to create the different forms that we see. But a much more common pattern is that it's the same old genes, just duplicated versions of them, or sometimes evolution of an orthologous copy. But the same genes gain new ability to bind, new targets, connected together in new genetic networks, and that results in different forms. And so to extend the analogy of toolkit genes, just like if you were to give the exact same toolkit to 2 different workers, right? So when I say toolkit now, I'm like real tools, like hammers and saws and screwdrivers and things. If you were going to give the exact same toolkit to 2 different people, based on how they use those tools, they might have very different outcomes of whatever they're building. So sometimes the same tools used in different ways, connected together in different ways, can yield very different outcomes. And so that's a good analogy for these Hox genes. The same tools in the fruit fly might lead to a very different outcome than those tools do in a mouse, but they have the same basic tools. Now, there are some important differences, and we'll highlight them in coming lectures. So remember, these toolkit genes establish a field. The initial field that's established is basically the entire body, right? The Hox genes help, well, even before that, the maternal genes established an anterior to posterior pattern, and then that pattern is elaborated as more and more downstream target genes are activated. And so the whole body is the initial field, and we can look at gene expression across that field by looking at labeling these genes as they are being used. So here is a secondary field. So we have segments that are already established, and then appendages developing within those segments. So we have secondary fields in the overall field, and you can even, depending on how you define them, you could get tertiary and quaternary, right? So that's the third level and fourth level developmental fields. And all of these will have different genes that are activating often the exact same axes. So we can look at an appendage and it has an anterior and a posterior axis that is defined by a different gene than the anterior to posterior axis of the entire organism. Okay? But just remember that. And so eventually, after we get down to the very, very last steps of all of these cascading genes activating one after another, we get differentiation of different cell types, all the way down to the very fine features. And so it's basically fields established within fields, until all the individual cells, more or less, are told what their exact cell fate is, and they coordinate with the cells around them through signals that are being sent back and forth by organizers to end up with the final organism. And there is some flexibility in this process, but it's remarkably robust. Now, robust means that there can be minor modifications and you still end up with a functional product. And so if we look at humans, for instance, like vein pattern, right? No two humans have the exact same vein pattern. Your veins vary slightly. That's why for some people it's easier to get IVs than others. You might notice sometimes quite substantial. So every once in a while, even large changes like heart, instead of being on the left side of the body, might be more displaced to the right side in some individuals. That's a small mutation. And many of these are just little variations in pathway without any real mutation in the DNA. There is some plasticity or some flexibility, if you will, to those developmental pathways. But despite that, it's remarkably how often, there's not often big major mistakes. They happen at a small but significant percentage, and many of those end up in unviable embryos. So spontaneous miscarriages, sometimes also called spontaneous abortions, occur in all organisms and are often the result of either a genetic mutation that is not survivable in the embryo, or just simply, this incredibly complex pathway, something going awry, either just by chance or because of some external force, a chemical, a temperature shock, or something like that. But eventually, all of these big fields get subdivided into smaller and smaller fields until we have individual cell type differentiation. And so in theory, we could look at all of the cells in an embryo, and of course, they're going to be increasing number of cells as we go through cellular division. One cell turns into 2, into 4, into 8. But in theory, we could look at all of them and map the cell fate. Now, for organisms like vertebrates with billions of cells, it is really tricky, and although we have a general idea, we don't have cell fate completely determined, and the farther we went down the pathway, the more levels of plasticity and variability there would be in cell fates, okay? But here's our model, and we've set this up. So we've got a precursor cell, and so the mother may create an RNA and differentially deposit that in the precursor cell, in the egg, right? Sometimes fertilization is actually one of the signals that differentiates one side from the other, where the sperm fertilizes the egg. That can serve as a prezygotic, not really maternal, but a fertilization signal and a prezygotic signal for how one side of the embryo is going to be different for the other. But in fruit flies, that gene we talked about, bicoid, among others. But bicoid is laid down in the anterior end, what will be the anterior end of the developing embryo. So when the cell divides, now we have two different daughter cells, because this one contains high levels of bicoid and this one contains low or no bicoid at all. And so now we have a differentiation. And that bicoid then might have differential. We may have, again, a gradient with differential. And so when we divide, we get different ones. Genes are turned on at one side versus the other. And so the zygotic genes begin to be activated in different areas. And when cellular division occurs, we end up with two different daughter cells. So notice by just a single, and often it's more complex than this, but a simple model is at each division, we can end up with doubling the number of differential cells. And so notice by the time we get to 8-cell stage down here, we have 8 different cells based on the transcription factors that are present in those cells. Okay, so it's a very simplistic model. Again, it's usually much more complex than this, but it's a good general overview for how this final cell type determination, after hundreds upon hundreds of cellular divisions and differential signaling by these diffusing morphogens, but how we end up with many different cell types. Okay, and all of this at the molecular level is gene regulation. Okay, so we've already defined what a gene is and looked at it. We're going to look at it in a little bit more detail. This is not a molecular biology class. This is really kind of in the realm of molecular biology. I only want you to understand the basics and realize that it's a very complex process, and each individual transcription factor needs to be studied, and many of them are being studied in model organisms, and we have pathways really worked out for some of them, but there's still a ton of work to be done. And particularly for organisms that are not model organisms, where we have not been able to study them in detail, there's still a lot of open questions. We're probably going to see many similarities to the model organisms because of the conservation of these pathways, but there are probably going to be new things, maybe a few new genes, but certainly many new pathways and new function for the same old genes that will help us to explain different body forms. Okay, so this is very diagrammatic. This is not intended to look exactly like you would see them at the molecular level if you were able to look at them under a microscope, which, by the way, you can a little bit, but they're really kind of on the edge of what we're able to look at effectively with microscopes. You can look all the way down to the single molecule level, or sorry, to the single atom now level, but those are very specialized circumstances, and in living cells, in functional situations, we have some pretty strong limitations in what we can do microscopically. Okay, so this little funky oval is meant to represent a transcription factor. It has bound to a region of DNA. We're going to call it an enhancer. If this transcription factor turns on the expression of the gene, it would be called an activator. I don't know why they didn't put activator there, but this would be an activator that turns on expression of the gene. Often there are other factors, sometimes even other proteins, that work together with the transcription factor. So it's kind of like a tag team where they need to be there together to fully activate the gene. But they do so by binding to the region of DNA called the cis-regulatory element, right? So let's actually put that up there just to remind you. The general term for a regulatory element, it's a region of DNA, and I'm just going to, let's pop a new one in here. The general term is a cis-regulatory element. Cis, again, referring to it is nearby, right next to the gene that it regulates. These regions of DNA can either be enhancers or silencers, depending on whether they bind a activator or a repressor. Okay, so I know the terminology is a little tricky. If you want to just write it down and have it to reference, or if you want to memorize it, that's great. Okay, so the regions of DNA that are the regulatory elements, if it binds to an activator and turns on gene expression, we call it an enhancer. If it binds to a repressor and turns off the levels of expression, right, down regulates the level of expression of the gene, then we call it a silencer. Now, this one here is labeled an enhancer, but in reality, it's a combination, both an enhancer and a silencer, because in the diagram, we have it able to bind to both a activator and a repressor. And so some regulatory elements can serve as either enhancers or silencers, and they do this by binding competitively. And so now we have even more variation and potential for differential control of the genes, because depending on the level of the activator in comparison to the repressor, we might have a different outcome. And if they're both there equally, we might get really little activity in the gene, maybe low levels of expression, not completely turned off, not really turned on strongly. But if we raise the level of the activator, gene expression would raise, and maybe even at a threshold point would suddenly dramatically come on and would remain on until levels of activators and repressors were then modulated by the surrounding organizers that are sending out those signals. Okay, so again, it's a complex thing, and understanding it fully is not within the scope of this class, but the general idea is important. And so what happens is when the correct activators are bound, they interact with the RNA polymerase complex, which is represented here in gene, and then they turn on and dramatically increase levels of gene expression. Whereas repressors will do the reverse. They will prevent RNA polymerase from binding to the gene, and you get no DNA expression. And so although there are some regulatory elements that can act as either enhancers or silencers, many of them are either or, and they work in a kind of a mosaic modular mechanism, meaning that there are often 3 or 4 or 5 of the exact same enhancers that can all bind the same activator. And then there might be a dozen of different repressors. And so depending on those, how many there are, and different types and different levels of these transcription factors that are present, we can have a huge wide range. So it allows for massive variability and even very fine control of levels of expression depending on the interaction of those transcription factors and all of those regulatory elements, right? So just know promoter, right? Or you can, if you want, you can say promoter complex, a group of proteins that will transcribe DNA, make the mRNAs for the genes, but they are controlled and regulated by the transcription factors binding to these regulatory elements. Okay? Oh, sorry. RNA polymerase is the complex, is the proteins that bind to the promoter. Sometimes a Tatabox is just one very common eukaryotic promoter. And they will bind to those, but only under the influence and under the direction of these transcription factors and their associated regulatory elements. Okay, so an enhancer again binds a activator, turns on expression, right? Sometimes there's also coactivators that either increase the ability of it to bind or are necessary and it won't bind without it, so that adds other levels of control. And then a repressor and a co-repressor bind to a silencer. I should probably put that in here also. Let's do that. So we'll make this one 6. And silencer is the regulatory element that binds to a repressor or a co-repressor. And remember, sometimes you can have elements that can act as both silencers and enhancers, depending on their ability to bind both activators and repressors. Okay, so terminology is a bit of review. Hopefully you already have it somewhat in your head. Review it, make sure you know it, or at least can get access to it to go over it, because it will be on exams. But other than that, we're not going to look at which specific ones bind and how they are impacted by competition with other ones. We'll just know that this is the general mechanism for regulating genes. Now, figuring out all of those details, although we aren't going to learn any specifically for this class, I do want you to understand one of the mechanisms that's a very important mechanism in us understanding how those work. Okay, so to study and figure out the impact of regulatory genes, we can do a number of genetic manipulations. This is one of the most important ones, the only one that we're really going to learn. So let's say we have 3 identified regulatory elements upstream of a gene of interest. Maybe this is the ILIS gene, the PAX6 gene that starts eye development in an organism. And we know where the promoter is, we got the coding region, so great, we've identified everything, but we don't know what the exact impact of these different regulatory elements is. Maybe we don't even know what transcription factors they bind to. We don't know if they act as enhancers or silencers, turning on, turning off gene expression, or maybe a little bit of both, right? And it can be, again, complex. So we can't study A only by itself, but by reducing this into its simplest components, we can begin at least to understand their action, and then maybe we need to put them back together and understand them in a integrated point of view. Okay, but first we can manipulate, we can isolate these DNA fragments, maybe in a eukaryotic organism like yeast, so it's still using eukaryotic mechanisms to promote, but is in a small single-celled organism that we can still study fairly easily. So we isolate these, and then we put them in front of what we call a reporter gene. Now, there are a number of reporter genes. For instance, beta-galactosidase is one that creates a sugar that we can identify with differential color change. But also, maybe the most common one nowadays is a fluorescent protein like GFP, or green fluorescent protein. And the reason we use reporter genes is because we can get a very easy, often immediate assay of levels of expression, right? So if we're looking at levels of expression of ILIS gene, we would have no way of just looking at that cell and seeing how much of that ILIS gene is present in that cell, right? The only way we could do it is through a Western blot, right? Some sort of a northern blot. I always get those mixed up. Let me bring it up here. Right, a northern blot is simply a laboratory procedure where we can look at, northerns are RNA. Maybe it was Western. These are kind of stupid names anyway because they don't refer directly to the process that's being done. So a Western blot is a laboratory procedure where we can look at what proteins are being expressed in a cell, and even we can do levels, although that's a little trickier, and there are more modern ways to do this also. But the point is that it's quite difficult to assay levels of expression for most genes. And so by making this construct where we put the 3 different regulatory elements upstream of a reporter gene, we can then add different transcription factors to these different, now genetically modified yeast organisms carrying this construct. And simply by assaying level of fluorescent, green fluorescent light that comes from them, we can have an initial measurement of expression, okay? And so these can even be done, although it's trickier, we can do them in model organisms like fly and mice, and we can look at them, and even in living cells, assay expressions by attaching these reporter proteins to our things of interest. And it's important that if you do this in these embryos that we don't create such a massive difference that it changes, kills, creates a vastly negative impact for the embryo, because then that's not a valid assay of what's really going on. So this is one way, and an important way you need to know about how we study regulatory elements. So all we do is isolate them, attach them to a reporter gene whose expression levels can be easily measured, and then measure them, and that begins to be a starting point that then we can use as we begin to look at them as they're integrated and in more complex manners, okay? And what we have found is that it's a very specific binding, that specific enhancers will only bind to a small subset, sometimes 1, 2, 3, but not very many transcription factors. So it's a very specific interaction between one transcription factor, sometimes 2 or 3, but a small number of transcription factors and a very specific enhancer. And if that binding serves to turn on the gene expression, we call the regulatory element an enhancer. I know I'm repeating myself a lot, but that's because it's important. So if that transcription factor binds to a regulatory element that increases expression, we call the regulatory element an enhancer, and we call the protein that binds to it, that transcription factor, is categorized as an activator. And of course, the reverse, if it downregulates expression, then we call the regulatory element a silencer, and the protein that binds to it is a repressor, okay? And as I mentioned earlier, these work in a very kind of modular way where we can add and subtract. Mutations can occur that create extra ones, and that might be a benefit, and so it's kept, and we get extra gene expression. They can be shuffled around and moved around, and there's quite a bit of flexibility and range of impact based on how these regulatory elements change. Now, the very cool thing about this is if the target gene for these regulatory elements is pleiotropic. So think about that. Hopefully in your head you immediately know what that means, but if not, let's review. Pleiotropy is where one gene has 2 or 3 or 4 jobs, right? Multiple jobs for a single gene. So think about that. If we have a pleiotropic gene, and almost all of these toolkit genes are pleiotropic, they have more than one function in the developing embryo. So if we mutate the protein itself, that almost certainly is going to be a negative bad mutation, right? Because it's going to impact all 5 genes, and a random mutation having an overall benefit impact on a gene with 5 jobs, the chances of that are very, very low. However, if we just change, move around, eliminate, add extra 1 or 2 regulatory elements, we may have impacted one of the jobs of the gene, whatever job is controlled by those regulatory elements, but all the other jobs are still completely functional. And so regulatory element evolution is a much more flexible and useful place for mutations and changes that are going to potentially result in a benefit because they don't have such a dramatic impact on all the jobs of a gene. And so this, to a very, very large degree, explains the conservation of these toolkit genes over incredible spans of time, right? The fact that we can have an eyeless gene and a PAX6 gene that can still cause eye development in 2 organisms separated by 600 million years or more of evolution shows a remarkable level of conservation. And so the gene itself has the same function. It's just the control elements that need to be in place to use it correctly. So if I took PAX6 from a mouse and put it into a Drosophila mutant that couldn't make eyes, I could get fly eyes as long as I had all of the fly regulatory elements in place. If I don't have those in place, right, if I have the mouse ones that are listening for mouse signals and responding to mouse signals, then it's not going to work. And despite the tool being the same, the ability to turn it on and interact with it correctly is very different. And so this is why really not all, but maybe 80-90% of the variation in evolution that we find is variation in regulatory elements and evolution of regulatory elements. There is some evolution of genes, particularly gene duplication, which then results initially in redundancy and eventually new functions. But the genes themselves often remain largely conserved over many, many hundreds of millions of years of evolution. So we are not, again, going to get into this at the level that we would in a molecular biology class. But I do want you to understand basically how networks work and how diagrams work. And so I'll probably do a little quick review of this and overview later in a different lecture. But upstream and downstream, the nice thing about these terms, the terminology here, is that they're fairly intuitive. Sometimes when I look at words that are used in biology, I wish I could go back and talk to the person who gave that name, like a northern blot or a western blot. Why couldn't they have called it something that was a little bit more intuitive about what was going on? But these are, luckily, okay? So upstream means a gene that occurs early in a sequence of one transcription factor impacting the expression of a gene and then another one and so on. So if it occurs prior, it's upstream. If it occurs later, in the time series, it's a downstream target gene. Necessary and sufficient. So necessary means that you absolutely have to have that transcription factor present or the gene cannot be turned on. Sufficient means that it can do it all by itself, that it doesn't need a coactivator, it doesn't need 2 different transcription factors bound to 2 different activators to do it. It can do it all by itself. And so if we're looking at expression of a single target gene, we could have transcription factors that are necessary and sufficient, that they can do it all by themselves and there's not an alternative pathway. We can have genes that are just necessary. Maybe there's there's no other way to do it, but not sufficient, right? So if I had, just to be very clear about this, if I had a activator that was necessary to turn on a gene, but it could not act by itself, it needed a coactivator, that would be a necessary gene. It would have to be present, but it wouldn't be sufficient because I would also need the coactivator being present. And maybe both of those, the activator and the coactivator, would be both necessary, and neither would be sufficient. Neither of them by themselves could turn on gene expression. So just realize that we can have all of the above. We can have transcription factors that are necessary, transcription factors that are sufficient, meaning they can do it all on their own, and even sometimes transcription factors that are both necessary and sufficient. That's the only one. Those are more simple models. Usually it's a little bit more complex than that. And then finally, when we are looking at direct versus indirect, direct is when it is a transcription factor itself. It itself directly impacts and turns on transcription. Indirect means it's a signaling molecule. So it doesn't do it itself, but it's part of the pathway that does it. So here in this diagram, we have this hierarchy where we have 2 regulators that feed into pathway A, and then this signal turns on binds to a receptor that then signals transduced till we finally get to a transcription factor. So this signal itself would be an indirect, right? So the regulator that's binding would be indirect because it doesn't bind to the DNA itself. It binds to a signaling molecule, a cellular receptor, okay? And then they have target genes. This target gene, if it interacts with another one, it would be direct if it interacts directly as a transcription factor. And the arrows, of course, represent the interactions. Now, in this diagram, I don't have it, but just one additional thing. In standard gene network diagrams, if it's an arrow, that indicates some sort of positive activation, meaning it's turning on gene expression. So this regulator binds to the pathway, and if that pathway then goes through, gene expression of the final targets, in this case 3 final targets, are upregulated. They get turned on. Sometimes there are feedback inhibition loops. If a gene is acting as a repressor, if it's turning off gene expression, I don't have a diagram here, but typically what you see is, well, I'll open it up here and show you what it looks like. So typically, rather than an arrow, we draw it like this, where there is a line, and then rather than an arrowhead at the end, we do just a perpendicular line, so something like this. All right, so rather than an arrow, I'm going to get them all. There we go. So you'd have something like that. Make that just, it's going to bother me. I know it's a little bit of a silly thing, but there we go. So that would be something that indicates a upstream gene that is a repressor, right? Whereas if we have an arrow instead, let me find the arrowhead. I don't do this all that frequency. There we go. Right? So if we have an arrowhead instead, like that, then that is an indication that the gene, the upstream gene that it's targeting this downstream gene, would be a activator. And again, both activators and repressors can be necessary and sufficient, or both, to turn on and or turn off gene expression. And so you're getting hopefully this picture of this kind of complex process, right? And we don't understand the great details of many gene networks and gene cascades. We understand them in basic principles and are still figuring out all of them. And so this becomes very, very important not only in development, right, but understanding these gene interactions is important for all cellular functions, because cells respond to change, are flexible depending on the genes are expressing. And so these are studied, these gene activation networks and gene control networks are studied in cancer and disease processes and normal regular cellular physiology, responding to outside stress, homeostasis. These are critical. This is the process of life, turning on, turning off genes, interacting differentially as necessary. And so this is not unique to EVO-DEVO, but it is essential to our understanding. And we're not going to get into the guts of these networks, but I do want to understand the basic principles that we've outlined here today.